Hi, my name is Iryna, and I am a PhD student at the University of Ottawa. And today I'll walk you through my poster entitled Real-Time Machine Learning Detection of Protein Contaminants During Mass Spectrometry Analysis of Affinity Purification Experiments. Proteins and their protein-protein interactions play a critical role in shaping cell behavior, which makes us really interested in having efficient techniques to study protein-protein interactions, especially in a high-throughput manner. And now the common method is using affinity purification experiments, which are coupled to mass spectrometry. So with affinity purification, you can choose a bait of interest and express it with a tag. You can then isolate the tagged protein along with all of its interactors and then identify them using mass spectrometry. Now, the issue is that mass spectrometry data acquisition has limitations for low abundance and transient interactions, and it typically collects redundant data about the same high abundance protein interactions at the expense of lower abundance interactions. Additionally, affinity purification experiments are susceptible to contamination, which means that more resources are wasted identifying biologically irrelevant interactions. But what if we could improve the way that the mass spectrometer acquires data in order to improve the sensitivity and also avoid identifying contaminants? So in my project, I am designing such a computational pipeline for affinity purification mass spectrometry experiments, which uses a real-time database search strategy to identify peptides and proteins live during an experiment. And then we can evaluate whether a putative protein interaction has enough data collected to confidently identify it, as well as whether it is a contaminant or not. And so if these conditions are true, we can exclude the instrument from collecting more data about the proteins and instead identify more biologically relevant interactions and those of lower abundance. So in the work that I get to share today, I would really like to focus on this step of how we can predict whether an interaction is a contaminant or not. In order to make this assessment, I begin with a set of negative control affinity purifications, which are experiments where only a tag is expressed. And these negative controls came from Bioplex, which is a large network of protein-protein interactions. With these negative controls, I use Bayesian inference to ultimately model the probability of seeing different abundances of a protein based on all of the observations from the negative controls. And so what's useful about these null distributions is we can use them to compare the abundance of interactions that we see in real experiments, and then even assign a probability of how likely a protein interaction is to be a contaminant or not. So in constructing these models, the first things that I looked at was a visual assessment of how well the probability distributions reflected the observed intensity or abundance of the proteins in the controls. So fortunately, we can see that the probability distributions in orange follow similar patterns to the real observations in green points. And so whether there is a wider distribution of intensities, a more narrow distribution, or even multiple peaks of intensities for a given protein, the Bayesian inference seems to reasonably capture these kinds of patterns. To qualitatively assess the modeling, I compared model performance based on the number of control experiments used. So looking between 5 and 50 randomly sampled controls, I built models and then predicted contaminant interactions from separate experiments. So this was repeated 50 times, with the average performance being shown here. To actually evaluate performance, I compared the number of identified interactions and an estimate of the false discovery rate. And so overall, we can see that models from all control set sizes could identify a reasonable number of interactions at biologically relevant false discovery rates. However, models between 10 and 25 controls really offered the best balance between reproducibility, the number of interactions, and also the false discovery rate. And so finally, I benchmarked our approach against SAINT, which is the standard filtering software in the field that detects contaminants, but can only do so once an experiment is completed. So using both SAINT and the Bayesian model to predict contaminants from the same experiment, I compared the false discovery rate and its coefficient of variation. So the graph shows the Bayesian model's performance with solid lines and SAINT's with dashed lines, and it's also color-coded by the control set size. And so what you may notice is that SAINT quickly hits a lower wall in the estimated false discovery rate, and it also has higher variation across replicates. So overall, this suggests that the false discovery rate is not well estimated with SAINT, and therefore not as informative for evaluating performance. And so we looked at a similar metric of false positive rate, which also reflects for us the proportions of false positive identifications. And here, both models identify a similar number of interactions at lower biologically relevant false positive rates. And additionally, the Bayesian models can achieve lower and more stringent false positive rates than SAINT. So it's very exciting for us to see that our Bayesian model has a comparable performance to the current gold standard in the field, because while SAINT is the leading tool, it cannot be used for real-time analyses. So this really gives us good confidence in the Bayesian model as a candidate to proceed further with and integrate into real-time analysis. As such, my next steps are to use this model in real-time simulations and on a mass spectrometer to guide data acquisition. But so far, we've really shown that with our algorithm, we have the capability to detect contaminant protein-protein interactions in real time, which is really unprecedented in the field. With that, I'd like to thank you for listening to my poster walkthrough. I'd also like to thank the members of my lab, my supervisor, and my funding sources for making this research possible. And of course, if you have any questions, please feel free to reach out. Thank you once again.